Let's start. Hello, everybody. Welcome to the VTLSS seminar this Friday. Today's speaker is Amy Pruden. She's the Thomas Rice Professor in the Department of Civil and Environmental Engineering. Amy specializes in applying microbial ecology principles to engineer water systems to address public health challenges. Amy obtained her bachelor in biology from the Department of Biological Sciences at the University of Cincinnati in Ohio, PhD in environmental science from the Department of Civil and Environmental Engineering at the University of Cincinnati as well, and then moved to her first academic position as Assistant Professor of Civil and Environmental Engineering at Colorado State University. She moved to the rank of Associate Professor at CSU as well. And then she finally realized that we are better than CSU and moved to Virginia Tech. She has been Associate Professor of Civil and Environmental Engineering first in August 2008, moved to the rank of Professor of Civil and Environmental Engineering in 2013. And in 2016, she became the Thomas Rice Professor of Engineering. I have to say that the record of accomplishments that I've seen on Amy's CV, wow, it makes me really envy in a good way. You're really a good model for me. She currently serves at the National Academies of Science Environmental Health Matters Initiative, which aims to bridge environmental and public health in addressing grand challenges. Serves at the on the National Academies of Science One Health Action Committee. She recently served on the National Academies of Science Committee on Management of Legionella in Building Water Systems, and is currently the Associate Editor of the ACS Journal Environmental Science and Technology. Let me tell you a little bit more about her. She has over 150 publications. Her H index is 50. Her CV was over 56 pages. I stopped the printer on page 56. Okay, I thought that the printer was going crazy and it was printing me 20 times the same CV. Then I realized it was only one part. She has received numerous awards, prizes, and recognitions in her lengthy career. I'm just going to mention a couple of them: the Career Award from the National Science Foundation, the Presidential Early Career Award in Science and Engineering, which is usually given to the most outstanding early career investigator among all the early career investigators. The Paul Bush Award for Water Environment Research from the Water Environment Research Foundation, the Dean's Award for Excellence in Research from Virginia Tech, the Jean and Leslie Douglas Pear Award given to organizations and to individuals who are dedicated to improving the life of others and to providing a sustainable earth for future generation. And this was given together with the Flint Water Study team. On top of that, she received the award for Outstanding Graduate Student Mentoring from Virginia Tech College of Engineering, and is a fellow of the International Water Association. Amy has an extensive record of funding that it makes me think that I need to go and write more grants. So without any further ado, please welcome Amy to today's talk. Thank you, Amy, for joining us.
Well, thank you, Carla, for the really kind introduction. It really is an honor to be the Life Sciences Seminar speaker today. I always enjoy attending when I can, and I know you get super high-powered speakers for this series. So thank you for having me. And I very much want to point out that, you know, the productivity that Carla highlighted, it's very much a team effort. And as I go through, I'll try to make it a point to highlight some of the contributions of my collaborators and students. So here's what I hope to chat with you all about a bit today. So as my title implies, I want to talk about water quality monitoring and advancing next generation DNA sequencing. So a big overarching question is, what can omics tell us about drivers of pathogen and antibiotic resistance gene proliferation in water systems? And can omics be adapted to inform risk assessment? So that's a big question for us in water quality. How clean is clean enough? Have I reduced the risk? What do I need to measure to inform that risk? And I work on a variety of water systems. I do work on wastewater and recycled waters. That's when you reuse the wastewater effluent for irrigation or even for drinking water, and then conventional drinking water. And I'll give some examples as I go along. I did want to highlight that it was a real honor to serve on this National Academies Committee where we looked at management of Legionella in water systems. And this was a team effort, a labor of love. This report came out last fall and really highlights many of the challenges when you're trying to merge, biology and engineering and public health to try to understand and stop the spread of disease caused by bacteria that grow in water systems. And there are lots of helpful recommendations in the report to try to stop the spread of Legionnaire's disease. One that I wanted to highlight had to do with the methods. And so Legionella are very tricky to study in water systems. So over here on the right, you can see the traditional media that's used to grow them. It's a charcoal yeast extract agar, and it might take several days to get results. And, not everything grows. It's not always specific. It also tends to mainly get what's called Legionella pneumophila serogroup 1, and that's the serogroup that's usually associated with Legionnaire's disease. But there's kind of a chicken and an egg situation where that's usually what we look for, so that's what we usually find. And for clinical testing, usually they use a urine antigen test because it's quick and easy, but it only tells you that you've got serogroup 1. And there's other species of Legionella. There's Legionella anissa, Longbeachiae, and Legionnaire's disease is caused by any bacteria in the genus Legionella. They vary in virulence, and that's something we're learning about, but maybe we need to know more about these other species. There's other serogroups, there's 15, there's other sequence types, and so there's just a lot of questions about what we should be looking for when we often advocate, we need to monitor Legionella in water systems. Now, culture is the gold standard, but there's growing popularity of methods like quantitative PCR because you can directly extract DNA, you can target a marker for that pathogen, and within a couple hours have quantitative measurements. And then where I'm really interested in going next is, what's the genomic and metagenomic potential? And so I think anyone at Virginia Tech has heard of the Flint water crisis, and I was part of the team with Mark Edwards that went in and tested the water, and the lead problem got a lot of publicity, but then it turned out that there was a Legionnaire's disease outbreak going on at the same time. And this is a publication that we put out that really nicely lays out the anomalies in water chemistry. So over here we have chloride, chlorine demand, temperature. So clearly during the water crisis, everything went haywire, right? And then in this bottom panel, we see that there was a Legionnaire's disease outbreak during that period as well. And this was a big deal. The way we deal with Legionnaire's disease, the Safe Drinking Water Act doesn't really address it. It tends to be very hodgepodge, managed in litigation. And so this was a big deal because it showed that a change in the municipal water quality could actually trigger a community-wide outbreak. And so we're looking now at, okay, what can we do at a federal and state level to try to prevent this from happening again? And In the abstract art for this publication, there's a nice illustration. So this was the water during the crisis at a hospital. And then this is 1 year later. So after the EPA, CDC, everybody went in and flushed and flushed the system, switched the water, got the corrosion control in place, the water cleared up. Now, as the microbiologist on the team, I really wanted to know which Legionella were causing the outbreak and, you know, who was in the water, can we match the water and clinical cases. And so this publication came out in the fall in Emerging Infectious Diseases. And so we were able to get clinical Legionella strains from the Michigan Department of Health and Human Services. And then we had managed to collect some Legionella from the water throughout Flint. And so we did the sera grouping and we did the sequence typing. So the sequence typing is you've got 7 alleles that you sequence and categorize them that way. And then we also did whole genome sequencing. And so what we found is there was a lot of diversity, even in the clinical samples. So those are shown in orange. You know, people weren't getting sick with the same strain, which is quite interesting. Like, to me, that tells me, you know, there's a systemic problem. The water chemistry shifted and the native Legionella, wherever it was in the system, bloomed and caused disease. And then when we did single nucleotide polymorphism analysis, we could kind of compare the strains. And so again, in orange, the clinical strains are kind of spread out. And then the water strains tended to be separate. So like this blue group was a lot of sera group 6 that was in hospital water. You know, none of the clinical strains that we had access to were sera group 6. Now, where there was some overlap was here, some water isolates and clinical isolates were quite similar. And you know, you got to be really careful here. Like, that doesn't mean it's exactly the same strain, you know. And then look here, the reference strain, Paris, as the name implies, that's from like across the Atlantic. That was very similar. And so we learned a lot. It's definitely interesting that, you know, there's such a diversity, but it definitely makes it hard to say like, okay, this source infected this patient. And then I also wanted to point out that we found a new species and strain of that species, Legionella taurinensis, and that became the subject of Connor Brown's master thesis. But larger point I'm making, and one that was really highlighted in the Academy's report, is that the Safe Drinking Water Act is not protective against Legionella. And this is in terms of where and how we place our controls, but also the methods that we use. So these are the things that are mentioned in the National Primary Drinking Water Regulations, and Legionella is mentioned, but this was written at a time we didn't understand that it grows in the distribution system. We were treating it like a contaminant that's in the source water. And so it's dealt with, they put treatment technique. And for all of these, Cryptosporidium, Giardia, viruses, they're very difficult to measure. So they don't expect at the treatment plant they're measuring these things and making sure they're removed. Instead, they say, okay, have this treatment technique. Usually, okay, filtration, disinfection, that will give you credits that you've dealt with this concern. The only thing we are required to measure are the coliforms, the fecal bacteria. And so how that works is kind of the you know, common sense approach that having poop in your water is bad. Okay, so these are tried and true traditional methods for organisms that grow in the feces of warm-blooded animals. They're, you know, rod-shaped, non-spore forming, ferment lactose, but few are actually pathogenic, right? So total coliforms is the big circle, fecal coliforms is a subset of that, E. coli is a subset of that, and then very few E. coli are actually pathogens. And this is a problem because Legionella, and I'd also like to highlight other opportunistic pathogens like Acinetobacter, Pseudomonas, Acanthamoeba, non-tuberculous mycobacteria. These are now the primary source of waterborne outbreak in the US and other developed countries. We've done a great job of dealing with fecal pathogens like Salmonella and E. coli and crypto. You know, it still pops up here and there, but what's really killing people are things like Legionnaire's disease. We've got 52,000 to 70,000 people contracting this per year and a 10% death rate. And then these other organisms are also quite problematic. And the fecal indicator bacteria approach, which is required by the Safe Drinking Water Act, has no correlation with these organisms. And that's because fecal bacteria, if they manage to penetrate the drinking water treatment plant, they still have a tendency to die in the distribution system because it's not their native environment. They like the intestines of warm-blooded animals. Whereas Legionella and these other opportunistic pathogens, this is their home. And they especially like it when the water crosses the property line into buildings where there's high surface area, there's stagnation, you know, the water's only flowing when you turn the faucet on, the temperature's warmer, the pipe material is reactive. So these things grow in the biofilms of the pipes. And then when you use the water and it's aerosolized, so for example, in a shower, they're spread by inhaling the water. So they tend to cause lung diseases, right? So both non-tuberculous mycobacteria and Legionella, they cause lung diseases. And again, it's the Safe Drinking Water Act, not the Safe Breathing Water Act. So that leaves us in a bit of a pickle. And if that doesn't give us enough to worry about, I do want to talk about antibiotic resistance too. So as an environmental engineer, my work has sought to advance thinking about antibiotic resistance as an environmental contaminant. So thinking about sources and pathways, how it spreads, how we can put barriers. Antibiotic resistance is a huge problem and it's not going away anytime soon. This report just came out from the CDC. So we're at about 35,000 deaths per year in the US. In India, it's estimated about 58,000 newborns die per year because of antibiotic resistance. And it spreads among bacteria because they can share genes, right? And so the World Health Organization and others have really been trying to get a global network going to combat the spread of antibiotic resistance, because I don't think we want to imagine going back to a world before we had life-saving antibiotics. And we began our work actually back when I was in Colorado, thinking about antibiotic resistance as a environmental contaminant and how it spreads in watersheds. And this report came out from the United Nations group that was focused on emerging issues of environmental concern. And they highlighted antibiotic resistance as their #1 emerging issue of environmental concern. And so this really brought the issue to the forefront. And I like this figure from the report really defined the concerns. So you have the majority of antibiotic use typically in livestock, right? And you know, we don't have wastewater treatment plants on farms, but we have some form of manure management to try to protect our waterways. We've got heavy use of antibiotics in aquaculture. And then of course, us human beings, when we use antibiotics, they're excreted, they go down the drain, they end up at a wastewater treatment plant, you know, and you know, our wastewater treatment plants are wonderful things that help protect the environment and public health, but they're not necessarily designed to deal with antibiotic resistance. And so you do get resistant bacteria and also some of the antibiotics themselves, it hasn't broken down, being emitted into the environment. And so there's a lot to think about here in terms of watershed protection, water treatment, and preventing exposure. And there's a lot of microbial ecology here too. So, what are the conditions that are going to attenuate versus select for resistant bacteria? And just a bit about the work we did in Colorado. So we've got the Rocky Mountains, and so we have a nice pristine source water. So this is the Puttive River at the top, flows from left to right, and it gets more impacted by urbanization and farm input. So we've got the livestock operations in yellow and wastewater treatment plants in red. And then Denver's down here, the South Platte flows up there and joins the Puttive east of Greeley. And so we saw, in fact, that if you measured the sulfonamide 1 antibiotic resistance gene, I'll call them ARGs, by qPCR, that there was almost a perfect correlation with the number of animal feeding operations and wastewater treatment plants upstream. And so this is where we've been working with thinking of the ARGs as the contaminants. This is a bit of a paradigm shift because usually we think about the pathogen as the contaminant, but because ARGs can be shared between bacteria and we've got the methods to directly measure the DNA, it makes sense to start thinking about how we can track the movement of DNA in the environment as a means of tracking antibiotic resistance. Now, I think we got a little lucky choosing the SUL1 gene. There were methods available at the time and we thought we'd look for it, but SUL1 is part of the operon of the class 1 integron, which has been elaborated upon by Michael Gilling's research group in Australia as really maybe the model genetic element we should be looking at and monitoring antibiotic resistance in the environment. They're really exclusive to clinical bacteria, and they tend to have this cassette with multiple resistance genes, and they're very highly associated with mobility. And so we tend to look at both the SUL1 gene and this class 1 integron integrase gene, INT-I1. So that's a little background. And so what I wanted to do is talk a little bit about some examples and then how we might move forward with these tools in the water industry. So I talked a bit about culture and whole genome sequencing and qPCR. Let's talk a little bit about metagenomics. And so that's where you extract DNA directly from the microbial community, do shotgun sequencing. And I think there's really a lot of potential here with the advent of next generation sequencing to tap into the power of metagenomics. And part of this is motivated by, we have 3 recent grants awarded by the Water Research Foundation. And this is a big deal that, they tend to be very conservative, like, okay, what does EPA require us to test, and kind of baby steps with new methods. It's kind of a big deal to be like, okay, we can do shotgun metagenomics now. And so they've funded these 3 projects. The first is linking various measures of antibiotic resistance in the environment with risk. The second one is specific about metagenomics and what value does it have for the water industry. Antibiotic resistance is part of that, but there's lots of other applications. And then this third one we're just launching is very specific, like, okay, we've got the water industry's attention that they should do something. What do they measure? And so we need some standards, we need guidelines when we say we want to monitor antibiotic resistance in the environment. And this is a table, and I don't expect you to take it all in, but kind of the idea here is we've got culture, qPCR, metagenomics, and we're trying to populate this table and get a sense of, what is each method telling us? Does it detect live organisms or not? Well, only culture can do that, right? So we have to infer viability using any DNA-based method. Potential to standardize, is it telling us anything about the microbial ecology, right? So I'd like to know if the ARGs are mobile and have potential to spread. And then of course, time, labor, cost, those are important things. So let's dive into some metagenomic examples here. So here we have it's supposed to be a cross section of a pipe with water flowing towards us, we'll say. And so it's showing that a pipe has a thick biofilm and that's this rich microbial habitat, and you've got thousands of microbial species, even in clean drinking water. I'm always humbled by how many different kinds of bacteria live in biofilms. And they're interacting, and the bulk water chemistry is affecting them. You know, does it have antibiotics in it? Does it have heavy metals? These are things that are known to put selection pressure on microbes. Are there plasmids? Are they engaging in transduction, conjugation? Is there evidence for natural transformation? So those are things we're really interested in from a microbial ecological standpoint. And with shotgun metagenomics, we can take the DNA from this whole microbial community and sequence it, right? And so these technologies have been moving fast. So we went from HiSeq to NextSeq, now we're doing NovaSeq. And then once we get all these sequences, then we can search public databases for virulence, markers, antibiotic resistance, and mobile genetic elements. So this first application is to wastewater treatment plants, and this is part of a National Science Foundation partnership in international research and education grant that Peter Vickeslund served as the PI on that. And this was a wonderful project where we had the opportunity to send students all over the world sample wastewater treatment plants. So we looked at the raw sewage and at various steps in treatment. So you usually settle the solids out of the sewage, then you aerate it in an activated sludge tank, stimulate biodegradation, settle that, and then often there's some disinfection and then discharge to a receiving environment, and then we try to sample upstream. So we standardized the whole monitoring process, and then we could compare across different countries. And this is an example of what we saw when we applied shotgun metagenomic sequencing to the sewage samples. So when we ranked them in terms of total antibiotic resistance genes to the 16S rRNA gene, we saw that the highest ranking was Hong Kong and India, and then the lowest was Sweden. And you can see here that the different colors correspond to different classes of antibiotic resistance genes that we were able to identify in the CARD database. And so, you know, to me, this is a really striking result. And really kind of what we were expecting. We know that Hong Kong and India don't require prescriptions for antibiotics, and you know, there's a lot of cases of new strains of multi-resistant superbugs coming out of India, whereas Sweden is the other end of the spectrum, where they're extremely proactive and controlling when it comes to use of antibiotics and trying to prevent the spread of resistance. So it was reflected in the sewage. And to me, this is very powerful and something that we should think about building on this, a global monitoring platform that could inform public health. Now, this next example is water reuse. So we live in a world where we have limited freshwater resources and they're dwindling, and it only makes sense to treat wastewater to the highest of standards so that we can reuse it, whether it's for irrigating agricultural fields, parks, or that's the case in this system at the top, which is a system in Florida. They use it for irrigation. And then the system at the bottom is one here in Virginia that we're exploring that's for potable reuse, recharging the Potomac aquifer, which is really oversubscribed. and is a critical drinking water source. And so this is part of the PhD work of my student Ishi, and just looking at the Florida system as an example. So various stages, so the sewage, you know, the primary treatment, activated sludge aeration tank, secondary treatment, they have a denitrification treatment, chlorination, and then we were able to look in their distribution system. We were interested in that because we know from our drinking water work that even really clean drinking water, the chlorine starts to decay, microbes start to grow in the biofilms. And so we definitely saw evidence of that, right? So we got low measurements right after chlorine, but they bumped up a little bit in the distribution system. Now, comparing the top versus the bottom is an example of where you have to make decisions of how to analyze the data. So relative abundance was where we normalized to the 16S rRNA gene as a housekeeping gene. This is nice because it helps you compare across samples, and it answers the question of, are there conditions that are selective for bacteria carrying resistance genes? And so when you look at the data this way, it suggests that, you know, the distribution system is an environment that's conducive to bacteria that have antibiotic resistance genes regrowing. But there aren't as many bacteria. So if you look at absolute abundance, those are at the lower end of the spectrum. And then culturing, a nice thing about this effort, it was funded by the CDC, and we brought in a collaborator, Jody Harwood, who's really good at culturing. It's a lot of work culturing. It's definitely easier to just extract DNA, give it to the sequencing facility, get data back. Culturing is a lot of work, and you're guessing the dilution, right? You don't know what's there. And so these dashed lines are the detection limit based on the dilutions we guessed. And these are 3 of the bacteria we were looking for: Klebsiella pneumoniae, Enterococcus, and E. coli. And then we put antibiotics in the medium that we were interested in: cefotaxime, vancomycin. So same thing, it's going through the treatment plant. And in general, they tended to go down and go below detection. So that's good. Now, not only is it a lot of work, but what these little boxes with the percents are telling us is when we did confirmation, what percent was actually what we thought it was. So you get these, you pay a lot of money for these media that are specific for these organisms. And then it turns out when you double check, it's a lot of other stuff's growing on the media. So that's a big problem. I think E. coli is a lot better formulated than some of the other things we were looking at in the wastewater. And so that's a lesson learned, that even when you go to the trouble of culturing, you've got to verify. And then we thought it was interesting here, we saw some evidence that some of the organisms could bump up in the distribution system. So that wasn't just something we saw in the DNA, but in the culture methods too. And now drinking water. And so a lot of my drinking water work is in collaboration with Mark Edwards. And this is an example where we wanted to compare drinking water with wastewater and recycled water. So this is work of my PhD student, Matt Blair, and in collaboration with Hampton Roads Sanitation District. We're interested in this concept that each water has a signature of its microbiome, and that might be some new dimension of characterizing water quality. And so it was interesting that the more you treated the wastewater to reach potable water standards, when you did the microbiome profiling of 16S genes, they tended to cluster together more. And so started to look like potable water and bottled water. And then the metagenomic screening and the work with Mark and Flint, you know, we looked at Legionella, and we did qPCR and metagenomics, and they correlated pretty well, so positive and significant correlation. But we were getting complaints from residents about a lot of other things, like there was a suspected Shigella outbreak and people were afraid it was in the water. What the evidence points to is that, you know, it's a hand washing issue and people were afraid to use the water. So that was a concern there. And so we tried really hard to see if it was in the water. And on the left, we did this metagenomic screen just to kind of see, okay, here are markers for all sorts of scary pathogens. And so according to this, there were some positive hits in the water, but then when we used the stringent PCR method, we couldn't find it. And so this is kind of a cautionary tale that, you know, I think metagenomic sequencing, at best it's a screen. We can say, okay, we found putative pathogens and we tend to hedge our language carefully. But you really, to know that these organisms are there for sure, you have to do follow-up testing. We saw the same thing for Stenotrophes multifida. That's one that we thought maybe was causing rashes that folks were reporting. But again, with more sensitive methods, we weren't detecting it. And the other thing is, we had a lot of data from other cities that weren't experiencing problems, and there really wasn't any obvious difference in the metagenomes.
Amy, can I ask you a quick question on that previous slide?
Sure.
When you collect your samples here, is this kind of a snapshot? I mean, you just go there and collect one time point, or this is, again, is a composite collection?
Sure. So we made a major trip in August 2015 when they were still using the Flint River. That's the far left. But we were only able to go to a few houses at that point. And then in October 2015, just before they switched to Detroit water, we tested again. And then March 2016 is when the water was recovering. I showed you the sink at the beginning of when the water cleared up. Mm-hmm. So, you know, we don't have a lot of time points, but for what we do, there's not a clear pattern here.
Yeah, thanks.
Now, I do think metagenomics has a lot of potential for pathogen screening. In this example, this is a totally different system, and we again saw had a really strong correlation with Legionella pneumophila measured by qPCR. But in this system, we were raising the temperature from left to right, and when the temperature was hot, they didn't overlap as well. Now, in this one on the right, it's just metagenomic data, and we wanted to look at other pathogens besides Legionella and besides Legionella. So we looked at Mycobacterium and Pseudomonas. And again, as it got hotter, here Legionella went down. We know that, like if you turn up the temperature on your water heater, that's a great way to kill off the Legionella, but it actually selected for the markers of these pathogens. So going forward, I think metagenomics has a lot of potential. The nice thing is you can look at so many pathogens at once and then try to optimize conditions for all of them, but then you've got to do follow-up work to make sure it's really a pathogen. Now, everything I've presented so far is all Illumina short read sequencing. We're doing a little bit with Nanopore now. I think it's very exciting. I think it's the future to do long reads. When we do that, you know, we could get up to 100 kilobase pairs, and then we know if the antibiotic resistance gene is on a mobile genetic element, we can identify bacterial hosts. And so that's a big quantum leap forward. And in theory, it's field deployable. And just some examples, Dongjuan applied Nanopore sequencing to some of the pyre samples. And we had this question, as you move from the sewage to the activated sludge, are you increasing resistant bacteria or decreasing? So there's some hypothesis that, you know, activated sludge, it's really actively growing microbes. We're trying to keep them in the exponential rate because they're biodegrading all the organic matter. Maybe they're closely clumped together and sharing antibiotic resistance genes by conjugation, and maybe phages are taking over, but that's not what we saw. So from Nanopore sequencing, the influence sludge network analysis, course, there were a lot of pathogens there and a lot of ARGs associated with pathogens. But then when you do the network analysis of the activated sludge, you have much fewer connections and less evidence of ARGs and pathogens. And then we could also see if the ARGs were in plasmids or chromosomes. So in these chord diagrams, on the left, we have classes of antibiotic resistance. The top is the influent, the bottom is activated sludge. And then on the right is whether it's in a plasmid, and then these different phylogenetic groups if it's on a chromosome. So proteobacteria, actinobacteria, etc. We were interested in how many were on plasmids. And so in general, the number on plasmids did go down in the activated sludge. This is just one example, but when we looked across several plants, that's what we saw. Okay, so finally, what do I tell the water industry? What do we monitor and why? Do we look at antibiotic resistant bacteria in general, or pathogens or ARGs, mobile genetic elements? You know, do we consider whether it's an imminent public health risk or something that's readily measurable? So part of the reason we look at coliforms is they're readily measurable. You know, we're gonna find them, and there's general modeling that shows that when they're absent, there's less disease risk. If we pick some really stringent, scary antibiotic resistance gene, we just might never find it, and then we don't get much information about what's happening. So that's where, collaborating with Carrie Hamilton at Arizona State University, she does quantitative microbial risk assessment. So we're trying to figure out how these different measures of antibiotic resistance fit into this framework. And then here at Virginia Tech, I've been working with Liqing Zhang, and her student Min helped develop what we call Metacompare. It's a way to measure resistome risk is what we're calling it. So it's the degree to which antibiotic resistance genes occur on mobile genetic elements and in pathogens. And then we have, you can plot it in 3D space or 2D space and compare. So in this validation, we showed that hospital effluent had the highest resistome risk, which is kind of what we expected. And we've started applying it to wastewater treatment plants. So this is my student Hania, monitored this wastewater treatment plant for a year and looked at the resistome risk score, and the influent in blue was always the highest and the final effluent was always the lowest, which is what we would hope. And then Ishi has also applied it to the recycled water systems, and similarly, the closer you are to the sewage influent, the higher the risk. And so this is a publicly available tool, Metacompare Web, and so folks are welcome to check this out. And one of the things you can do is actually zoom in and visualize the contigs and the annotations of the ARGs, MGEs, and pathogens. And then nano ARG is something that Liqing student Gustavo developed. And so it's configured for nanopore sequences and looking at ARGs, MGEs, also metal resistance genes can be of interest, and you can put in modules for other functional genes as well. Now it doesn't have to be nanopore sequences, it can be assembled Illumina sequences. And then Lenny Heath has been a really key collaborator here as well. And so we have a USDA funded project called AgroSeq. And here what we're super excited about is to get people to share metadata. So not just, you know, this metagenomic DNA sequencing data in a vacuum, but tell us where it came from, what kind of livestock, what kind of antibiotics are being used, is it soil, is it water, what about the soil? Just try to get as much information that's associated with these metagenomes as possible, and then we can start supporting people who share their data in comparative studies, and then also modeling and identifying, you know, where are the critical control points where we need to focus our mitigation efforts. And then in the Water Research Foundation project, the one that's focused on risk, Nick Ashbolt is part of that project as well. And so he's collaborating with us and a third risk assessment approach, which is Bayes nets. And it's a probabilistic approach where you kind of map out all these sources and pathways and put probabilities here. And the nice thing about this is you can take into account microbial ecological processes like conjugation and gene sharing and whatnot. And then finally, we're super excited that this grant just got funded by the National Science Foundation, and it was in the VT News this week if you're paying attention. And Li Ching is the PI here, and I have to hand it to her. Like when she came up with this idea a year ago, like based on our prior work, like, hey, let's equip wastewater treatment plants for public health monitoring, and you know, we can have real-time metagenomic measures, and we can put the data in the cloud and have Ali help with that. You know, we can see when antibiotic resistance is emerging before it's a problem, and we can tell doctors, we can report to public health agencies, you know, here's antibiotic resistance that has emerged, and maybe you should switch which antibiotics you're using. And so it seemed pretty crazy a year ago, but I was thrilled. And then now, you know, COVID's happened, and it's, everybody wants to monitor sewage now as a public health resource. And I think that's great. This is my hope. We'll come on the other side of the pandemic and really tap into sewage monitoring as a public health resource. And it takes us back in time. You know, we think as a humanity we're evolving, but we keep bumping up against many of the same problems. And I recently read this book, The Ghost Map. I highly recommend it. So John Snow is credited with being the father of modern epidemiology. It was just, you know, started simple, mapping out where people are getting sick, where people are dying, figuring out what's the common thread. And in this case, it was drinking water from the Broad Street pump. And so, yes, in the future, I'm stoked about monitoring sewage as a resource to protect public health. And, you know, the trick with these molecular methods, they're very powerful and they're a two-edged sword. I do worry about people over-interpreting the information. So we cannot directly measure live organisms. And with most of the time, the way we're using them, we can't even say they're definitely pathogens there. We have to be cautious, and we have to be aware of these limitations and how we interpret the data. But that being said, I think there's a ton of potential here. And so here are some of the tools we're working on, and I'd be very glad to entertain any questions. And thanks again for having me today.
Thank you very much, Amy. Awesome talk, really super interesting. We have a couple of questions. I'm going to start with actually one from me. I just have a curiosity. So you report your findings to where? I mean, beyond the publications, to the department, and I mean to some, you know, environmental department, and they just go and take some action. I mean, what happened after you sample the places, you identified all these antibiotic resistant bacteria? What is the follow-up on this?
That's a good question. So that, I guess that's what we're working towards. Like, part of the problem is the system we live in under the Safe Drinking Water Act. It's not required to monitor any of these things. It's only required to monitor fecal coliforms. And so it gets difficult to break through that. And I understand, because, you know, water utilities don't want another problem. They don't want to scare people. And this is research, you know, we haven't fully defined the risks. But that's part of why we've been pretty public with sharing our data. I mean, we publish it in scientific journals, but you know, I make it a point to go to conferences attended by engineers and water researchers, people like practitioners in the field, and it's gotten their attention. And actually the state of California has been really taken notice, because they've got severe water shortages, and they do have citizens that are concerned, like paying attention and concerned. And so they've actually they've given money to the Water Research Foundation to fund these projects. And so the state of California, they have their own guidelines for determining that water is safe when you use it for reuse. They tend to mirror like the drinking water guidelines, right? Like the easiest thing to do is say, okay, we're following federal drinking water guidelines, so it should be fine, right? But the federal drinking water guidelines are, they fall short for things like antibiotic resistance, right? And there's different risks with water reusing wastewater. So they've put out a few reports where they're starting to be a little more forward thinking, starting to embrace, like they call it contaminants of emerging concern. Like, okay, we'll start keeping an eye on this and keeping record of the data until we know more. And so I think that's where we are right now, is in a situation of, just trying to collect these data and kind of do a surveillance and try to get a sense of what they mean and when we should be concerned.
Well, thanks. We have a couple of questions, and I don't want to miss anybody here. Paul Marek says, fascinating talk, thank you. Thinking about biofilms and the different pipe compositions, would copper versus PEX or PEX versus another composition be better for reducing biofilms in drinking water pipes?
That is an excellent question, and it's unfortunately not so simple. We just submitted a paper for review that covers the different materials encountered in plumbing, and copper and PEX are at the top of the list. And of simplistic sense, you'd think, okay, copper we know is an antimicrobial, right? Okay. But it's not necessarily releasing that much copper in the water. With any given water chemistry, the copper is usually not doing much. And if it is being released to the water, then you worry maybe the water is too corrosive and you're gonna corrode your pipes. So it's definitely not as simple as just getting copper pipe. PEX pipe, so that's a plastic that's flexible and heat tolerant. So like if you have hot water plumbing, it can tolerate that. But that, it's relatively inert. It does leach organic carbon into the water. But both, they both get plenty of biofilm. So when you consider the list of what controls like Legionella, for example, in water, the material is pretty low on the list. It starts with temperature being high enough to kill the pathogens, having disinfectant in the water like chlorine, having good hydraulics, so you're flushing the system, and then, materials sort of comes into play there, but is a minor factor.
There's a follow-up question from Xiaoming Yang regarding biofilms in water pipes. How prevalent are they in pipes for municipal water?
Oh my goodness, they're everywhere. Like if you dig up a water main, it's pretty gross. I mean, they're thick. Okay. Anywhere you have water in a surface, you will have a biofilm. And so my philosophy is we can't eradicate biofilms, but we can shape them, right? So if we understand the conditions that select for good bacteria versus bad, like we can, find the right water chemistry and conditions that the bacteria that grow are benign and not antibiotic resistant or pathogens. And most bacteria are benign, so.
Okay, we have another question from Daniela Cimini. You talk about the link between antibiotic prescription practices between Hong Kong, India, and Sweden to explain the difference in levels of antibiotic resistance. But how much does population density play into this?
Very insightful question, Daniela. So we have not yet published the paper with the beautiful figure of the sewage antibiotic resistance genes, where they're highest in India and lowest in Sweden. And part of it is because when you try to get into the weeds of what is actually leading to that pattern, right? There's the population density, socioeconomics. We've measured antibiotics in the sewage, so we have that data at least. One thing we found is that the antibiotics that we measure do not relate to what's reported. So, you know, most countries have, they report their sales data, right? So that's one indicator of how many antibiotics are going into the environment is the antibiotic sales. And so to us, it's eye-opening that you can't trust a lot of the data that's out there. And, you know, to the extent that we can actually measure things, so we measured the antibiotics for ourselves, we trust that data better. And there's other things, markers you can measure in the sewage to have a sense of population density. Like we all excrete certain phages, like crass phage, and pepper mottle virus is a plant virus we excrete. And so we can look at the density of those and have some indicator of population density and whatnot. So, but it's very tricky. And that's something now that we've been collaborating with Carla and trying to do some of this with COVID, it's really opened my eyes to how many things you have to take into account.
Okay, we have two more questions. This talk brought a lot of comments and questions. Have you done any investigations to correlate number of ARGs in wastewater with ARGs in clinical isolates from the concurrent region time period?
From which region?
From concurrent, corresponding.
Oh, I see.
It's a cool idea.
Yeah, so I mean the CDC data from the water reuse treatment trains does that a little bit. So we looked, I showed you 3 resistant pathogens, Klebsiella, E. coli, and I think Acinetobacter. We did some others, Pseudomonas and MRSA. So, you know, that's a long list of pathogens to try to test. So those are ones we cultured, and then we have the metagenomic data. And so that's, my student Ishi is, hopefully that'll be a chapter in her dissertation, is comparing the measurements of these clinical resistant bacteria to the metagenomics. But that's what we're measuring in the sewage now. To say to the rest of the population, we're doing a little bit of that. So Hania, the wastewater treatment plant she studied was in Virginia, and Virginia does publish these antibiograms of resistance in a given region. And so we're doing some comparison there, but we'd like to do more.
I'm gonna ask you to answer one more question, and I promise I'm gonna let you go. This is from Ben Uruch. Have you heard of any studies that try to correlate the presence of antibiotic resistance of bacteria in the water to public health? For instance, do these areas see higher rates of hospitalizations for bacterial infections? That's a pretty cool question.
Yeah, that's $1 million question, and
yeah,
I wish I had a better answer. Part of it's we don't have good surveillance to try to match those numbers up, and so most of what I hear is kind of anecdotal. There are studies that have at least done the modeling. Like there's a study, the Thames River and concentrations of resistant E. coli that can cause urinary tract infections, and showing that yes, the levels in the river are at the correct dose to cause these infections, and you know, have some evidence in the population. We tried to do a study of a city in Arizona that did irrigation with recycled water, and unfortunately the data were a mess, and I hope we can figure something out there. But yeah, we need more work in that area to link that together. We need more data. So
okay, well, thank you very much for your presentation. I'm pretty sure that everybody really enjoyed it, because I get a lot of really nice comments out of the screen that you probably don't receive, but but thank you very much, and I'll see you around.
All right, thank you everybody.
See you guys. I'll see you next Friday. Amy, I have a quick question.
I'm still here.
Okay, I read the ghost map. as well. It's a fantastic book. Is that picture that you have there of the well, the real well in Broad Street?
I think so.
You know,
Is it not the right? You
because
know, I think
everything
it was
that well, right? At the end, I mean, they discovered that it's in that well, but I thought that that well was.
You're right. They moved it.
Yeah.
Yeah, I remember hearing that it's not actually there anymore.
Oh, okay. It was
I
curiosity.
think, yeah, and then, but it is like there's a memorial there. So, but I think it's like
Not the
walk
original,
away from where it
very
really
first
happened.
one, not the
I
original.
think so. Yeah.
Okay, just curiosity. Okay, take care.
All right.
See ya. Bye.